Welcome students. This is for Diploma in Pharmacy students. Not only for Diploma in Pharmacy students, this class will be more useful for degree students and even scholars who are doing some research. Maybe a simple class of chemical test and a preliminary phytochemical investigation can be performed by means of listening to this class. And my information to the Diploma in Pharmacy students: listen to this class and note it down all the chemical tests, and you should submit this report on coming Monday. Very important. That is, I will give you the instruction clearly in your group. You should submit clearly. You have to listen to this class and you have to write a summary also. At least the names of the chemical test listed out in a page, and before that all the tests should be clearly explained. I need a summary as well as complete chemical test details have to be written and it should be submitted. All the first year Diploma in Pharmacy students, I hope you have understood what is my assignment. This class we are going to discuss about qualitative, quantitative, qualitative. This is qualitative identification test for preliminary phytochemical screening and identification test for, according to the syllabus, 2 chapters have been covered in this one lecture actually. Preliminary test for phytochemical screening and identification test for starting from carbohydrates, proteins and amino acids, alkaloids, tannins, phenolic compounds, flavonoids, glycosides, steroids and terpenoids. Usually they are present in glycosides. So steroid and terpenoids under glycosides we have seen the chemical test, and volatile oils. There are still many specific chemical tests are there, but this is generally if you want to have a preliminary phytochemical investigation, and I have given almost all the test detailing, giving that what is carbohydrate, what is protein, what is alkaloid, what is tannin, what is phenolic compounds, what is flavonoids, what is glycosides, what are its slight types, and the chemical test, and the steroids and terpenoids and volatile oil, so that the importance of the chemical test will be more useful and more easy for you to write. If there is any questions, short questions, if they ask, it will be easy for you to write clearly. So now first we will start with the qualitative test for carbohydrate. Everybody might have studied in most of the classes, even most of the other courses also, including biochemistry also. You might have studied very clearly. Carbohydrates, what are they? Just a simple definition I am giving. Carbohydrates are formed by green plants from carbon dioxide and water during the process of photosynthesis in plants. They serve as energy source and an essential structural component in organisms, in plants, in all the organisms, and even it is a very essential primary metabolite for animals also. So we are first starting with the simple monosaccharide D-glucose. There are, you know that you might have studied more detail about D-glucose is an aldose, ketose, like that you might have differentiated and studied the carbohydrates. So fructose is also a monosaccharide, but it is a ketone derivative. Sucrose, disaccharide example. And you can see polysaccharides. I have taken an example, starch, which you might have studied detailedly about the two parts of water soluble and water insoluble parts of starch, like amylose, amylopectin. You can see the linkage of C6 linkage and the C2 linkages of the glucose molecule of the monosaccharides and disaccharides, how they are glucose molecules are seen as in polysaccharides. So just a definition. Now we will go to the chemical test as per the topic. Detection of carbohydrates. First one is Molish test. Plant extract plus or add 2 drops of alcohol with naphthol and 1 ml or 1 milliliter of concentrated sulfuric acid along the walls of the test tube. A ring is formed. A ring. What color? A ring in violet indicates the presence of carbohydrate. Violet color. Violet indicates there should be a space. A ring of violet color is formed in the test tube. The next one is Fehling's test. Plant extract added with 1 ml of each Fehling solution A and B, Fehling solution B, and then boil the test tube in a water bath. A red precipitate indicates the presence of carbohydrate. We should know what is Fehling solution A. Fehling solution A is very simple. It's a blue colored aqueous solution of copper sulfate. And this is that solution. And a Fehling solution B is a colorless aqueous solution of potassium sodium tartrate. Potassium sodium tartrate, that is KNaC4H4O6, and 4 water molecules is present in that molecule. Also known as, it's also what type of salt? It is called Rochely salt. That is called the potassium sodium tartrate. It is made very strong of alkali by sodium hydroxide. Already potassium is itself an alkali. It's made more strong alkali by sodium hydroxide. That's why it is potassium sodium tartrate. So Fehling solution A and B is added, and then you are getting the red precipitate while heating in the water bath. Number 3 chemical test for carbohydrate is Barfoot's test. 1 ml of Barfoot's reagent plus plant extract heated for 2 minutes. A red precipitate due to the presence of monosaccharide indicates the presence of carbohydrate, and it's a red precipitate. So you can see this. You should know what is Barfoot's reagent. Barfoot's reagent is a mixture of ethanolic acetic acid, ethanolic acetic acid with copper acetate is added to the test tube solution and boiled. That is the Barfoot reagent, means ethanolic acetic acid with copper. That means copper acetate. So this, the result you can see, it gives a color, a red precipitate actually. You can see the test tubes, series of test tubes are shown here. Very clearly the presence of monosaccharides, xylose, glucose, fructose, they give red color. But you see the lactose and sucrose, they are disaccharides, they never give results in Barford or Barford test. The chemical test can be seen by the test tube which I have shown in the picture. Then now Benedict's test is a very interesting test, and it is very commonly used for diabetic patients for urine analysis. So test for plants, what you will do, 0.5 ml of Benedict reagent plus the plant extract, boil for 2 minutes. You will get a green or yellow or red according to the presence of quantity of sugar or quantity of the carbohydrate in the plant extract. The color will be changed, green, yellow or red. Indicates the presence of carbohydrate. So you see this, when you add only the reagent before heating, it will be in a blue color. If it contain a less quantity, it will become green color. If it is slightly more quantity of carbohydrate, it will become an orange or yellow color. It will become a brick red color as shown in the last fourth test tube, so that you have more content of sugar molecules or more content of carbohydrate is present in your plant extract. Benedict solution is a chemical reagent which is made up of complex mixture of sodium citrate, sodium carbonate and pentahydrate of copper sulfate. So this you have to remember. Simply writing the scientist name and reagent will not be sufficient. What the chemical is present in that particular reagent should also be mentioned. Now, qualitative tests for proteins and amino acids. What is amino acid? They are building blocks of proteins. Proteins are long chain of amino acids. Our body has thousands of different proteins that each have important jobs. Same thing in plants also. They also have the amino acids as their building blocks of their structure. This is L-amino acid. So amino acids, when they join together, they become peptide, and peptide will join and they will make protein. So amino acids of the plants play a major role in a multitude of plant reaction, or multitude of plant reaction, not multitude, multitude of plant reactions, contributing greatly to their correct development and growth, as well as to the production of hormones. Now, detection of proteins and amino acids. What all the chemical tests? Number 1, Biuret test. Biuret test is in that 1 drop of 2% copper sulfate solution added with the plant extract and add 1 ml of 95% ethanol and add potassium hydroxide pellets. A pink colored solution is formed in the ethanolic layer. You know that ethanolic layer, you will get a pink color, indicates the presence of proteins. Number 2, the second test, Millen's test. Few drops of Millen's reagent plus plant extract, a white precipitate is formed, indicating the presence of proteins. Millen's reagent consists of metal nitrate of mercury and mercuric nitrate that is dissolved in concentrated nitric acid, so that you will get metal nitrate of mercury. That is the Millen's reagent. Now we are number 3, is ninhydrin. Ninhydrin test, plant extract and a few drops of 2% ninhydrin solution. Is then the test tube is kept in a hot water bath for approximately 5 minutes. After a few minutes, a blue or a violet, slightly violet color indicates the presence of amino acid, amine and protein groups. And number 8, xanthoprotic test. Xanthoprotic test in which few drops of concentrated nitric acid added with the plant extract, a yellow color solution indicates the presence of proteins. So you can perform this test very interestingly. Now we are coming to the qualitative test for alkaloids. So we should know what is alkaloids before going to the chemical test. Just the definition. Alkaloids are nitrogen containing compounds that are structured in a heterocyclic ring structure with nitrogen. Here carbon, nitrogen will be there sometimes inside the ring, sometimes outside the ring also. Carbon, hydrogen will be present, hydrocarbon compounds. In addition to that, oxygen and sulfur in occasion may be there. Usually nitrogen is already nitrogen containing compound. So first example, ephedrine, where you can see the nitrogen is outside the ring actually. In the side chain, ephedrine structure everybody knows is an alkaloid. And nicotine, you can see it contains 2 nitrogen, one in the pyridine 6-membered ring structure and pyrrolidine 5-membered structure also we have one N-methyl group inside the ring structure, nicotine. Morphine also you can see an N-methyl group inside the ring structure. And you can see strychnine, they all have 2 nitrogens inside the ring, a complex structure which you can see very clearly is an indole structure. Indole alkaloids, quinoline alkaloids like quinine, anti-malarial drug, cinchona constituents, main constituent, quinine is also an alkaloid of quinoline derivative. You can see the ring structure, presence of nitrogen in the naphthalene ring structure, and even a N in the 5-membered side chain inside the ring. So this is about, at least the student will be knowing nitrogen containing basic compound is an alkaloid. It has various medicinal activity which is present as a secondary metabolite in plant sources. And detection of alkaloids, when you see first test, drag and drafts, or you can call as Kraut's or Kraut's, K-R-A-U-T, Kraut's test, in which 1 to 2 ml of drag and draft reagent with plant extract, a precipitate that is red-brown is formed. But you should know what is drag and draft reagent. Is bismuth subnitrate with potassium iodide in 2 molar nitric acid. Otherwise, you can say it is a solution of sodium bismuth iodide. Very simple, you can say what is a drag and draft reagent means solution of potassium bismuth iodide. Hager's test number 2 for alkaloids. 1 to 2 ml of Hager's reagent is added with plant extract, a whitish precipitate that is creamy, slightly creamy, yellowish whitish precipitate you have obtained. Hager's reagent means picric acid. It itself already it is a dark yellowish color which gives a precipitate, and the color is different in a qualitative test that you know very well. When you do in the lab you can understand. Number 3, Meyer's, or it can be called as Bertrand's, or it can be called as Walser's test. Usually Mayer's test we used to say. 1 or 2 drops of Mayer's reagent plus plant extract will give you a cream yellow precipitate. Mayer's reagent means potassium mercuric iodide solution. And number 4, Wagner's test, in which 1 to 2 drops of Wagner's reagent plus plant extract inside the test tube walls when you add, a precipitate of brownish red can be obtained. What is Wagner's reagent means? Iodine in potassium iodide, iodine in potassium iodide. So when you make yourself a hints of this reagent, you can remember all the test very easily and the color. Examples of plant alkaloids: quinine comes from cinchona tree bark. Caffeine comes from tea and coffee leaves. And morphine comes from opium poppy. Atropine in belladonna leaves. Ergometrin is present in ergot. Reserpine is present in Rauwolfia. So there are still many alkaloids of secondary metabolites which are used as a medicinal purpose, which has many therapeutic activity, which are many formulation also available for various ailments. Here particularly we have some specific alkaloidal test. Previously what we have seen is general alkaloid test. Now specific alkaloid test also they used to ask. For cinchona calisaya or cinchona ledgerania or cinchona officinalis, there are many species which you might have studied in theory. Family also you may have seen, it is Rubiaceae. So you can see that there is a thalloquine test specifically for cinchona. The presence of quinine will give this test very clear result. Quinine, as we have discussed, is a quinoline alkaloid. The nitrogen is present in the quinoline ring structure, and also the nitrogen is present in the pyrrolidine ring structure. Quinine. Take the extract of cinchona powder and add 1 or 2 drops of dilute sulfuric acid and 1 ml of water. Add bromine water dropwise. It's very dangerous. Bromine water we should add very carefully. It should not split in your hand or in your body. You should use gloves, or you should be very careful when you are using bromine water. Add bromine water dropwise until the solution acquires a permanent yellow color like this. And 1 ml of dilute ammonia solution, it will turn to an emerald red color as shown in the picture. It means it contains quinine. It is cinchona. Is produced, then it indicates the presence of cinchona and the constituents of quinine. And now we are going to another test of specific tropic acid, that is tropane, tropic acid, not tropic, tropic acid esters of tropic acid, which are example like atropine, scopolamine, gives an intense purple color in the Vitali-Morin reaction. It is Vitali-Morin test they used to ask direct question, allowing to distinguish them from the other tropane alkaloids. It is mainly example belladonna, Atropa belladonna, Solanaceae family, and it was a very good interesting plant, and it has many alkaloid from Egyptian. It was very long back, 3000 years back it was a medicinal use, and it was called as beautiful lady also, the name synonym, because it was used for even as an poisoning for many rivals, and beautifying the eye Even the extract of the leaf was poured in the eyes to make dilate. You know very well, it is an anticholinergic drug. It can dilate the pupil and makes attractive for the females. But now we will go to the test. It is a specific test for Vitali Morin's test. If you have to take a china dish or porcelain dish, instead of say china dish, you can say porcelain dish, can be used for this test for atropine and scopolamine. See the structure of the tropic acid when it is having an ester formation, COOH formation, which gives the esters of tropic acid. You take the sample, the plant extract of the belladonna powder you can take, extract or powder, in a porcelain dish, add fuming, that means concentrated fuming nitric acid, evaporate to dryness, heating in the water bath, and dissolve the residue in, add acetone and dissolve the residue, because once you finish, make it dried, it will become a dry residue, and dissolve that residue with the use of acetone and methanolic solution of potassium hydroxide. When you add methanolic solution of potassium hydroxide, you will get a violet color, which indicates the presence of atropine or scopolamine, which are the tropic acid esters, which are present in this Solanaceae family of Atropa belladonna, and even it is in Datura stramonium and Datura metyl. They all contain these constituents. Now we are coming to the another alkaloid specific test for ergotamine, which is present in ergot. Very interesting constituents, ergotamine. Ergotamine is used for migraine, used for abortifying agent it is. Very interesting activity. It is a Clavicis purpurea actually. Clavicis purpurea is the source of this plant. It's not a part of the plant, it is developed by a microorganism especially. Spore develops and which contain this active principle of ergotamine, ergocristin. There are many ergot alkaloids are present. A test which can identify specific test is Van Hertz test or Van Hertz test, URK'(S), Van Hertz test. Here, solution of ergot alkaloids is added with Van Hertz reagent. What is this Van Hertz URK'(S) reagent? It is very simple. Para-dimethyl amino benzaldehyde in 15% of sulfuric acid is the reagent, containing little trace of ferric chloride. If you add ferric chloride after adding this component in sulfuric acid, deep blue color is formed as shown in the picture. We have many alkaloids as I shown: ergotamine, ergocristin, ergosine, ergorenine or ergomine, and ergocryptin is also present by means of slight differences in their substitution. You should know that lysergic acid, LSD, the primitive test used to detect LSD, lysergic acid, which is more active as an nervous active stimulant, and CLSD, and related to ergot alkaloids, as well as based on the drug compounds. The reagent contains, as we have told, it is para-dimethyl amino benzaldehyde in a solution with sulfuric acid, and hydrochloric acid is often referred to as para-dimethyl amino benzaldehyde test or PDMAB test. Now we are to talk about the next alkaloid, myroxide test, that is caffeine. Comes from tea and caffeine leaves. They are having a structure related to xanthine derivative actually. They also have purine basic alkaloid. purine derivatives, they are not giving the common test which we have seen, that is common alkaloidal test which we have seen, the 4 tests like drag and draft reagent, Wagers, Meyer's reagents will not give any positive result for this caffeine. So caffeine should have a specific test. So they prepared this Mirroxide test. The Mirroxide test is an analytical technique to identify the presence of caffeine, which is an xanthine derivative, or you can say it's a purine alkaloids, purine derivatives and other purine derivatives in a sample. These compounds do not respond to the common alkaloid identification test such as drag and draft reagent, Wagers reagent or Meyer's reagent. They will not give any result. But in this test of Mirroxide test, the crude drug, the tea powder or a extract of a coffee is mixed with a tiny amount of potassium chlorate in a porcelain dish and a drop of hydrochloric acid. The sample is then evaporated to dryness as we did in the previous test, and resulting residue is exposed to ammonia vapor, or you can add even ammonium hydroxide, or ammonium hydroxide can be added, or the vapor can be shown. Produce a pink color, which is due to the formation of Mirroxide. The Mirroxide is called as purplish. So you will get a purple color. You can see this picture which I have shown. 1 ml of alkaloid solution means just you are taking the extract from the belladonna and evaporating, and you are evaporating to dryness and cool, and you are adding concentrated HCl and a few drops of potassium chlorate, and again you can heat it and make it in a residue, will become yellowish color and red color, and when you show few fumes or few drops of ammonium hydroxide, ammonia vapor, when it just show the vapor itself, you will get the purple color which I shown there. So this ammonium picrate, ammonium purpurate, that is why purple color, purpurate, ammonium purpurate is called as Mirroxide. This ammonium purpurate appears purple in a pure state. It shows that the sample contains caffeine. And quality test for phenolic compounds now. That alkaloids, we have seen general test and we have seen some alkaloids for specific test also according to your syllabus. There are many, but still I just conclude here. And now we can go to the qualitative test for phenolic compounds. What is phenolic compound? Phenol, aromatic OH group should be there in an aromaticism. Phenolic compounds possess a common chemical structure, am I right? Comprising an aromatic ring, a benzene ring, one or more hydroxyl substitutes that can be divided into several classes. There are many classes even these phenolic compounds. The main groups of phenolic compounds include flavonoids, phenolic acids, tannins, stilbenyls, and ligands. Flavonoids, very interesting therapeutic activity, constituent, phytochemical constituent present in most of the plants, gives interesting therapeutic activity, therapeutic effect. Luteolin, this is a structure as we said, aromatism with many OH groups. Luteolin, phenolic acid, just a phenol, aromatic phenol with OH, with having a COOH acid, 2 substitution, phenolic acid. Tannins, this is actually we know that hydrolysable tannins, aromatic ring structure which has many OH and also one acid group, tannins. We have separate test for tannins also, flavonoids also. But apart from that, they all comes under phenolic compounds. General test for phenolic compounds. Still beans, also same, having 3 OH group as a basic structure. Ligands, most of them are with CH2OH, alcoholic groups, aromatic groups with OH, as well as aliphatic alcoholic groups also present, ligands. They also comes under phenolic compounds. Now we are coming to the detection of phenolic compounds, in which is a common phenolic test, in which iodine test means few drops of dilute iodine solution added with plant extract, a translucent red color, which indicates the presence of phenolic compound. Ferric chloride test, few drops of 5% ferric chloride solution added with plant extract, dark green, bluish black color is formed. This indicates the presence of phenolic compound. Gelatin test, 1% of gelatin solution plus 10% of sodium chloride add with the plant extract is dissolved with 5 ml of distilled water. A white precipitate is formed, which indicates the presence of phenolic compounds. These all are just for identifying phenolic compounds. Now we can go for specific test for tannins, for flavonoids separately. Now we are coming to the flavonoids, which is also comes under in the phenolic compound. Qualitative test for flavonoids. Flavones are one of the important subgroups of flavonoids. They are coming in the phenolic compounds. Flavonoids are widely present in leaves, flowers, and fruits and as glycosides. Celery, parsley, red pepper, chamomile, mint, and ginkgo biloba are among the major sources of flavones, having more therapeutic activities. Celery means it's also a leaf, as a salad decorating leaf, like parsley, which is obtained from the coriander leaf. Other things we know very well. These all are having more flavonoids like luteolin, apigenin, and tangeritin, belonging to the subclasses of flavonoids. Like luteolin, we have discussed about the presence of phenolic group in aromatic system with an lactone ring. Apigenin is also the same structure, slightly varies in their position. And when we come to tangeritin, is also having methyl group is attached with all the OH group. It's also an flavonone having a flavonoids effect. This is the basic structure of the flavonoids. They were all flavonols, chalcones, anthocyanins, flavonols, isoflavones, and flavones. So there are different flavonoids they have identified by the research, which is more important in the therapeutic activity. That's why I just shown. They are also comes under as an phenolic compounds present as a secondary metabolites in plants. Now detection of flavonoids, many times they have asked question. In that you should know Shinoda's test. Plant extract, add a few fragments of magnesium ribbon. Magnesium tannins, magnesium ribbon, small magnesium ribbon, and concentrated hydrochloric acid, and add dropwise. By dropwise you have to add concentrated hydrochloric acid. Pink Scarlet color as shown in the picture appears after few minutes and indicates the presence of flavonoids. Sodium hydroxide test, in which 1 ml of plant extract, few drops of dilute sodium hydroxide solution added, an intense yellow color is absorbed as shown in the picture, and it becomes colorless on addition of few drops of dilute acid, which indicates the presence of flavonoids. Now lead acetate test, in which to the plant aqueous extract add 10% of lead acetate solution. Yellow precipitate is formed, which indicates the presence of flavonoids as shown in the picture. Now qualitative test for tannins, they also comes under phenolic compounds. But what is tannins? Tannins are naturally occurring compounds, organic compound possessing nitrogen, free polyphenols of high molecular weight. In the class itself I told you should remember they are astringent. They precipitate proteins. So tannins have two subdivisions. Hydrolyzable tannin, just now we have seen this structure, and condensable tannin having a multi-structure, that is condensed tannin, and this is hydrolyzable tannin. These two tannins are present as in classes. Detection of tannins, first one is gelatin test. Plant extract, aqueous solution of gelatin and sodium chloride are added. A white bluffy colored precipitate is formed, indicating the presence of tannin. White bluffy color. And gold beater skin test. They used to ask, what is this gold beater test? What is gold beater? It is just a skin which is taken from ox. That's why gold beaters test. A small piece of gold beater skin, that is membrane, prepared from the intestine, not skin, intestine of an ox, is soaked in 20% of hydrochloric acid, rinsed with distilled water and placed in a solution of tannin for 5 minutes. The skin piece is washed with dilute water again and kept in a solution of ferrous sulfate. A brown or a black color is produced on the skin. This is due to the presence of tannin. So this is what you have to remember about the intestinal part, a membrane which is prepared from an ox. That should be remembered. And number 3, phenazone test. A mixture of aqueous extract of a drug, that means a tannin containing drug, and a sodium acid phosphate is heated and cooled, and then it is filtered. The filter rate that the solution, a solution of phenoxone is added now to the filtrate. The filtrate which you collected, you have to keep in the test tube, and you add a solution of phenazone, not phenoxone, phenazone. A bulk colored precipitate is formed, indicates the presence of tannin as shown in the picture. A matchstick test, catechin test, is an interesting trick. Take a matchstick and dip that matchstick without that phosphorus. Dip the matchstick in an aqueous plant extract. Dry near the burner and moistened with concentrated hydrochloric acid. On warming near the flame, you can see the color change of the stick. The matchstick wood turns to pink. First you have to dip as shown in the picture, and second you have to go for the flame for drying, and then you can see the color of the matchstick. it turns to pink or red due to the formation of fluoroglucinol. Indicates the presence of tannin. Indicates the presence of tannin. And the test number 5 for the tannin is chlorogenic acid test. An extract of chlorogenic acid containing drug is treated with aqueous ammonia. A green color is formed on exposed to air, indicating the presence of tannins. The last test for tannin is vanillin hydrochloride test. They are asking this specifically. Sample solution of the plant extract added with vanillin hydrochloric acid reagent. What is this vanillin hydrochloric acid reagent? Take vanillin 1 gram, alcohol 10 ml and concentrated hydrochloric acid 10 ml. That is the vanillin hydrochloride reagent. When you add this, immediately a pink color or a red color is formed due to the presence of fluoroglucinol. Indicate the presence of tannins. Now, qualitative test for glycosides. Very interesting. Glycosides, you should know what is glycosides. Glycosides are the compounds with organic molecules having attached with glucose or mono-oligosaccharide units. Otherwise, you might have studied in detail aglycan and glycan part, which has linkage of O linkage, C linkage, N linkage. There are many linkages of glycosides you might have studied. But anyhow, there is an aglycan and a glycan part in a glycosidal linkage. Generally, aqueous or alcoholic extract of a crude drug are tested for specific region for presence of various types of glycosides. Glycosides can be in several classes. So we should know the classes. First one is anthraquinone glycosides, cardiac glycosides, saponin glycosides, steroid and triterpenoid glycosides, cyanophoric glycosides, coumarin glycosides, flavonoid glycosides. This also comes under glycosides, but flavonoids we have discussed in the phenolic containing compounds also. We have discussed the Shinoda's test. Everything comes to the same, to the flavonoid glycosides. Though I am not going to discuss about the flavonoid glycosides in this junction. Already we have discussed. So now we will come to the chemical test for anthraquinone glycosides. This is a structure of anthraquinone. First the test is bone tracker test. This anthraquinone having a linkage of 2 glucose molecules of O linkage. You can see the structure clearly. The chemical structure which is shown O linkage. You see the arrow, 2 O linkage which with the glucose molecules. The aglycan and the glycan are linked with an O linkage. Simple explanation to remember. Because they have another one test which is called modified bone tracker test. So that will make you to understand. This is test for the glycoside or an anthraquinone glycoside which are having O linkage glycosides. 1 gram of drug with 5 to 10 ml of dilute hydrochloric acid boil on water bath for 10 minutes and filter. The filter was extracted with a non-aqueous solvent like carbon tetrachloride or benzene. And when you extract equal amount of, after that taking out the extraction, because carbon tetrachloride is not miscible with dilute hydrochloric acid or the drug which is aqueous, because this is a non-aqueous solvent, you can separate to the another test tube an equal amount of ammonia solution and shake well. Formation of pink or a red color in the ammonical layer. Why? It is due to the presence of anthraquinone moiety or anthraquinone glycoside. So the senosides is one example. There are senoside A, senoside B are there. Cascara species which has O glycoside, they will give this result. But there is a modified bone tracker test, just not much like a structure which I have shown here. Cascarosides alloin which is present in aloes. They have C glycosides and O glycosides are also present as shown in the picture. This is O glycoside with O linkage with a glucose, and this is C linkage with a glucose. You can see the picture, the arrow came and shown you. So here only a change. 1 gram of the drug you take, or the plant drug powder or drug, add 5 ml of dilute hydrochloric acid followed by 5 ml of ferric chloride, which we have not done there, and boil for 10 minutes in a water bath and filter, and this filtrate you can go for extracting with carbon tetrachloride, and then transfer this carbon tetrachloride to the another test tube or a benzene, an equal volume of ammonia solution, ammonium hydroxide or ammonia solution, forming a pink color, red color due to the presence of anthraquinone moiety. So this is used for C type of anthraquinone glycosides. C type of anthraquinone glycosides as alloins as found in aloe species, C glycosides which are barbaloin actually, and they are also called as deoxybarbaloin, which can be called as chrysaloin. Barbaloin, chrysaloin, they are C glycosides. Chemical test for cardiac glycosides. So in which we have cardiac glycosides, some example which we studied like foxglove, digitalis purpurea leaves, very interesting beautiful flower, and Strophanthus combe seeds, they are also cardiac glycosides. Squill, they also contain cardiac glycosides. Urangenia maritima bulbs, it is underground bulbs. They are used as a rodenticide actually. The number 1 test is Killer-Kilani test, is very interesting important test. To the alcoholic extract of the drug, equal volume of water and 0.5 ml of strong acetic or not acetic, strong lead acetate solution was added. Shake and filtered. The filtrate was extracted with an equal volume of chloroform. The chloroform extract, because it is a non-aqueous, as evaporated to dryness, and a residue in a porcelain dish can be obtained, was dissolved with 3 ml of glacial acetic acid, followed by addition of few drops of ferric chloride solution. The resulting solution was transferred to a test tube containing, this resulting solution will be transferred to a test tube containing 2 ml of concentrated sulfuric acid. The test is little longer, so you should do in such a way. Reddish brown layer is formed, which turns bluish green actually. You see, change to reddish brown layer turns to bluish green after standing due to the presence of digitoxose. An interesting testing, even you can do by a test tube also, or even a china dish up to the drying, and then you can go for the test tube also, it's possible to do. Very interesting testing too for cardiac glycoside, especially digitoxose, which is present in digitalis purpurea. Legal test. To the alcoholic extract of a drug, equal volume of water, 0.5 ml of strong lead acetate solution was added. Shake and filtered. You can filter. The filtrate was extracted with equal volume of chloroform, extracting with a non-aqueous, that is aqueous, so using a chloroform, and this chloroform extract was evaporated to dryness as we did in the previous, in a porcelain dish we can do. The residue was dissolved in 2 ml of pyridine now, and sodium nitroprusside 2 ml are added, and followed by addition of sodium hydroxide to an alkaline. To make it alkaline, you can add sodium hydroxide. You will get a pink color. Formation of pink color as shown, the arrow shows. It shows the presence of cardiac glycosides or an A-glycan moiety. Number 3, Baljet test. thick section of leaf of digitalis, leaf directly, are a part of the drug which a cardioglycoside containing drug, strophanthus or squill, when dipped in a sodium picrate solution, it forms yellow to orange color, the presence of a glycan of glycoside. 3,5-dinitrobenzoic acid test to alcoholic solution of the drug, cardioglycoside drug, digitalis purpurea or digitalis lenata, or you can say strophanthus combe, or you can say even the squill. Alcoholic solution of the drug, few drops of sodium hydroxide followed by 2% solution of 3,5-dinitrobenzoic acid was added. Formation of a pink color as shown in the picture indicates the presence of cardioglycosides. Even before going to the next step, you should know that there are in cardioglycosides also there is two types are there, cardenolides and bufenolides. Bufenolides are present in like squill it contain. Cardenolides usually present in digalactose, and they are having the structural difference. If you see, the 5-membered lactone ring is present in cardenolides, 6-membered lactone ring will be present in bufenolides. That is the change which we can see. They are having but biological and their therapeutic activities also different in their applications. So you can see this 5-membered ring structure lactone ring and 6-membered ring structure lactone ring. By arrow I have just denoted you to note it down. Chemical test for saponin glycoside now. Examples for saponin glycosides, glyceritin, which is present in licorice, adi madiram, we know that, very much interested pharmacological use, and it is used more in Ayurveda and Siddha and Unani, our traditional practices, or our alternative medicine or traditional medicine practice, they are using it. And you can see this glyceritin present in licorice and there is quilaic acid. Quilaic acid present in quillia, or you can write even diastenin present in Dioscorea. There are many examples. They are having a structure like linkage of glycoside linkage, but the structure you can see is a lengthy ring structure which gives, they are saponin, and the saponin which will cause forming and lysis in blood also. So the first test itself, hemolysis test, in which a drop of blood on a slide you have to keep and mix with few drops of the sample aqueous saponin solution, means the sample containing saponin glycoside. What happens? The RBC becomes ruptured and it shows the presence of saponin. They are poisonous actually. And foam test, you can see 1 gram of the drug is added to 10 to 20 ml of water, and if you shake for few minutes, formation of frothing which persists for 60 to 120 seconds. It shows the presence of saponin. Now we are coming to the next one, steroid and terpenoid glycosides. Steroids and test, they may ask always. Steroids in phytochemical investigation, steroids are present in most of the plant sources. So it needs for you to chemical test. You can see that is triterpenoids, examples like olenin, uracane, lupane and damarine. There are many tests, specific monoterpene, diterpene like that, but it is terpenoids common. And steroid, colostane, spirostane, furostane, cardenolide, these all comes in the steroidal structures. Example. Chemical test for steroid and terpenoidal glycosides. Number 1, Lieberman-Bouchard test, name reaction. How you can perform? Alcoholic extract of the drug was evaporated to dryness and extracted with chloroform. Add a few drops of acetic anhydride followed by concentrated sulfuric acid from sides and the walls of the test tube to the chloroform extract. So formation of violet to blue color ring as shown in the picture. At the junction of the two liquid, you can have a non-aqueous and aqueous liquid. As shown in the picture, indicates the presence of steroid moiety. Interesting test. And number 2, Shalquoxy test. Here, alcoholic extract of drug was evaporated to dryness and extracted with chloroform and a concentrated sulfuric acid is added from sides of the test tube to that non-aqueous extract which you made, chloroform extract. Formation of yellow color ring at the junction of the two liquids as shown in the picture and turns red after 2 minutes. It will turns red after 2 minutes, indicating the presence of steroidal moiety. Now comes the chemical. It is continuing because you have 3 slides for the steroid and triterpenoid glycosides. The third one is antimony trichloride test. Here, alcoholic extract of drug was evaporated to dryness and extracted with chloroform. Add saturated solution of antimony trichloride in chloroform containing 20% of acetic anhydride. Formation of pink color on heating indicates the presence of steroid and triterpenoid. You can see that pink color. And number 4, trichloroacetic acid test. Here, alcoholic extract of drug containing triterpenes on addition of saturated solution of trichloroacetic acid forms a red colored precipitate as shown in the picture. And this is the 5th test for steroids and triterpenoids, glycoside. Tetranitromethane test. Here, addition of tetranitromethane forms an yellow color with an unsaturated steroids and terpenes. And number 6, you can see Zimmermann test. Zimmermann test also addition of metadinitrobenzene solution to a alcoholic solution of the drug, that is a sample containing alkali. On heating, it forms violet color. Is presence of keto steroids. So now the next glycoside which you want to know is chemical test for coumarin glycosides. Coumarin, they contain HCN actually. They will give green and then it becomes yellow. To the concentrated alcoholic extract of a drug, that means a drug containing coumarin glycosides or a plant extract, few drops of alcoholic ferric chloride solution, formation of deep green as shown in the picture, and turns to yellow when you addition of concentrated nitric acid indicates the presence of coumarin. Fluorescent test is also there for coumarin. Mix alcoholic extract of the drug, of the plant extract with 1 normal sodium hydroxide solution. Develop a blue fluorescent as shown in the picture. Blue fluorescent resonance presence of coumarin. Cyanophoric glycoside. In cyanophoric glycoside only hydrogen cyanide. Cyano, not in the previous one, coumarin. Here, sodium picric test. Moisten the powder which you want to keep in the test tube or in the bottle. You have to moisten the powder and keep in the flask. It's an interesting test which can be performed in the lab. Powder of the drug which will be moistened in the water in a conical flask or in a bottle or some provided testing bottle, Borosil, bigger bottle with a cork. a few drops of concentrated sulfuric acid. As shown in the picture, cork and keep a trap one filter paper. A filter paper is trapped, which is impregnated. Already the filter paper is dipped or impregnated with sodium picrate solution, followed by a sodium carbonate solution also on the neck of the flask using a cork. You can see a cork is kept. So where the formation of brick red color, you see the first one shows the very brick red color is formed due to the volatile hydrogen cyanide which comes from the cyanophoric glycoside if it is present in the drug and takes place, so that you can see the piece of the filter paper becomes red in color. Even the bottle can be used, or this type of specially prepared test tubes, bottles can be used. In conical flask with a cork, bigger cork can also be used for this experiment. Now the last one which I want to discuss in this class is test for volatile oil. You know volatile oil also. They produce temporary stains actually because they are volatile. A smear on a paper, filter paper, or in a tissue paper or a cloth, the stain disappears after some time because the oil volatiles. Second test is upon addition of Sudan 3 alcoholic solution to the thin section of volatile oil containing drug, red color is formed. Sudan 3 means what is? It is an lysochrome, fat soluble dye. It is a diazo dye. It is structurally related to asobenzene. This you should know Sudan 3 means. And when a few drops of tincture which is prepared from alkana plant added to the section of a drug, for example any volatile drug like a clove, or you can say if you see eucalyptus, if you take a piece of the drug, red color develops indicating the presence of volatile oil. This is the plant of just the reagent which is prepared from this plant. This is called the alkana plant which is prepared, a tincture is prepared, the plant of alkana tinctoria, which gives that extract and which can be applied to the volatile oil containing drug will become red in color. So that is about the test, and there are many specific tests which are there for individually for carbohydrates also. There for like acacia, tragacanth, and even resins. Individual tests there are many, but this one is for preliminary test. It is very much useful for you to read and to listen to this class. Write your assignments very clearly, the diploma students, and submit to my office on coming Monday. Thank you for listening this class.